Okay, the second gram-positive bacteria we want to introduce is the Streptococcus. In this word, strepto, okay, it means easily bent or twisted like a chain. So it's like here. This is a real gram-stained picture of Streptococcus. You can see they are grouping in a chain, and they are gram-positive. So this Streptococcus. The bacteria can be classified or categorized into different groups based on different criteria. First, if we check their colony morphology and their hemolytic activity, whether they can lyse red blood cells or not, and we can group them into 3 different types. Or if you check their serologic specificity of the cell wall substances, so the cell wall antigen, they can be classified into 17 different groups. That is, we call the Lancefield antigen. So that's a group-specific substance on the cell wall. First, the hemolysis. 3 different types of hemolysis. Okay. You understand this word hemolysis? What's the meaning of hemo? Blood, right? Here we are talking about the red blood cell. Okay, they lyse the red blood cell. And lysis. So that's, we use a blood agar, which contains the intact, the whole red blood cell. Okay. Then we culture the bacteria on the surface. Okay. And the first type of hemolysis is called beta hemolysis. Beta hemolysis is the complete disruption of the erythrocyte, the red blood cell, okay, with clearing of the blood around the bacterial growth. So if the bacteria grow on the surface to form either colonies or bacterial lawn, and surrounding the bacterial growth, here you can see there is no red blood cells. It's complete clearance of red blood cell. This type of lysis we call beta hemolysis. So the one we learned before, the Streptococcus. So the Staphylococcus is also beta hemolysis. And the second type is alpha hemolysis. This is due to the incomplete lysis of red blood cells. And the hemoglobin is released and then is reduced to some green pigment. Okay, so that during the alpha hemolysis, you will see the part of the agar is stained with green color. So this is alpha hemolysis. While the third one, we call the gamma hemolysis. Actually, it's not hemolytic. Actually, it does not cause any hemolysis. It's here. This is a gamma hemolysis. There is no actual hemolysis here. So 3 different types of hemolysis. And in this group, Streptococcus, some of them are beta hemolysis, some are alpha hemolysis. Well, you don't have to remember all these results, all this data, but some important pathogen we are introducing in detail, you should know their hemolytic activity. Okay, if we check their serologic types, that's the group-specific substance on the cell wall or the Lancefield antigen. Based on that, the bacteria can be grouped into 17 different groups. That's due to the C carbohydrate on the cell wall, on the cell surface. Okay, in each group, it's named with letter A, B, C, D, A to H, K to U, so totally 17 groups. In each group, there might be more than one species, one or more than one species. For human, Group A, B, and D are more important pathogens. And in all these groups, Group A contributes to 90% of human pathogenic streptococci. Okay, so the streptococcal infection in human, 90% is due to Group A. You don't have to remember this table, okay? Just to give you some information. Okay, the first pathogen we want to introduce is Streptococcus pyogenes. Streptococcus pyogenes. This is the typical infection in the throat. It causes throat infection. Sometimes when you have fever and you also have sore throat, and you go to the doctor, well, the doctor would think that, well, this might be a common cold, or it might be a pyogen infection, Streptococcus pyogen infection. They can run a quick test, use a cotton swab to take some specimen and run a test to tell you in a few minutes that whether it's a bacterial infection or not. Has anyone done that before? When you go to see a doctor? Coronavirus, not virus, bacteria. They check the presence of antigen. So it will give you the results in several minutes with a strip. Okay, let's talk about the pathogens. No, this is not a cold agent. The cold, there's something I want to tell you. All the common colds are due to viral infection. There are over 200 different types of viruses which cause a common cold. Well, you can recover by yourself if you have a common cold, but this one is a bacterial infection. You need to use antibiotic to kill the bacteria. Okay. So the S pathogens. First, we check their morphology and how to identify them. They are typical organisms. They are gram-positive, as you can see, and they form chains, long chains, or some isolates might show in pairs, and they do produce capsules. So there are capsule structures surrounding the cell, and the major component in the capsule is the hyaluronic acid. It's a polysaccharide. It does not produce catalase, so it's catalase negative. Remember when we learned Staphylococcus, it's catalase positive, right? Last time we learned that. So yeah, when we learn these specific pathogens, you want to compare them. So that will help you to remember the properties. And when we culture the bacteria on the blood agar, they would form the discoid colonies. And it's beta hemolytic. So on blood agar, it clears the red blood cells completely. So this is beta hemolytic. And the most important antigen produced by this bacteria is called the M protein. M protein is a hair-like projection, hair-like protein. It's a protein molecule, okay? Hair-like protein molecule from the cell wall to the extracellular space. And this substance is a major virulence factor of aspergillus. And I didn't show you the picture. It's an electron microscope picture of a whole aspergillus cell. And you can see the cell is hairy, a lot of M protein on the surface. It's like, imagine it's like the tennis ball, okay? A lot of hair. And this protein can be, let me see. The protein can be 60 nanometer long. So 60 nanometer long. Function of this M protein is first adhere to the host cell for adhesion, or that it plays some role in the evasion of the immune system. Let's see, function of M protein. It's a major virulence factor. It can also produce several important enzymes. The first enzyme is called streptokinase. Streptokinase. If you heard the name of kinase, what's the meaning of kinase? Kinase means that it can activate other molecules, right? So this kinase can transform plasminogen into plasmin. We know that plasmin is the active enzyme. It's a protein enzyme. So the proteolytic enzyme which can degrade fibrin. And we know that fibrin is the insoluble protein in the blood clot, right? So that plasmin can degrade fibrin in the blood clot. And if the bacteria is locked in the blood clot, it can escape, right? So that allowing the bacteria to escape from the blood clot. So streptokinase, even though it does not directly help the bacteria to escape, but it can activate plasminogen to form plasmin, and plasmin can degrade the fibrin. I know we have a lot of material, and if you understand the mechanism, if you understand how different molecules work, it's easy to remember. Okay, you don't have to, well, understand the information without understanding it. That will be difficult. Okay, that's streptokinase, the first enzyme. It also produces an enzyme called streptodorinase. Streptodorinase. The full name of this enzyme is Streptococcus. Okay, D means deoxy. Okay, dor means deoxy. Okay, R here means ribo. N means nucleus. So this is a streptococcal deoxyribonuclease. So actually it's a DNA. It degrades DNA molecules. Previously we mentioned that our immune system developed different mechanisms to fight against bacterial infection. And one of the mechanisms is that neutrophils are able to release their DNA from the nucleus to the extracellular matrix to form the neutrophil extracellular trap, NET, so that in the extracellular trap, it can bind the bacteria and kill them. So this way, release the DNA to the extracellular, trap the bacteria and kill them. But meanwhile, the bacteria evolved ability to secrete their DNAs. Okay, DNAs, to degrade DNA, cut DNA into small pieces. The small pieces DNA is more fluidity. It's not viscous anymore, so that the bacteria can escape from the trap. It's DNAs. It can also produce hyaluronidase. We learned this before. So that's the enzyme which can hydrolyze hyaluronic acid, which is the important component in the connective tissue. So that can also help the bacteria to escape and evade. The bacteria are also able to produce a lot of pyogenic exotoxins, and they mediate pyrogenicity, such as the disease we'll talk about in later, the scarlet fever and also the toxic shock syndrome are due to the production of exotoxins. This bacteria produces two different types of hemolysins. Again, hemo, the red blood cell, lysins, it's able to lyse the red blood cells. Hemolysins. The first one is streptolysin O, okay, type O. This streptolysin is able to stimulate the immune system to produce antibody. So it's antigenic. It can produce, the immune system can produce anti-streptolysin O, the antibody, okay, to bind to the antigen. But streptolysin O is not stable in the presence of oxygen. So once there is presence of oxygen, this type of O will be inactivated. So that's the two major properties of this one. Another one is called streptolysin S, second type. Okay, this lysin S is oxygen stable. So it's stable in the presence of oxygen, but it's not antigenic. It cannot stimulate the immune system to produce antibodies. And the beta hemolysis we saw on the blood agar, the halo, the complete clearing of the red blood cell is due to the streptolysin S. Okay. Just keep in mind ASO this term, because later we'll talk about how to check the presence of ASO in human blood. We will talk about that in detail later. Clinical findings. The disease due to the Aspergillus infection. There are 4 different types of disease due to different mechanism. The first type is due to the invasion by the bacteria and also their products. So the group A bacteria invasion and also their products. Or that some disease is due to the local infection by the Aspergillus and also their byproducts. The third type is due to the invasive type A infection, but it's mainly due to their exotoxins, like the toxic shock syndrome and scarlet fever. Both of them are due to the exotoxin production. And the last one is the post-infection disease. It's not due to the bacterial infection directly, not due to the bacterial cells directly, but during the bacterial infection, the immune system produces a large amount of antibodies, and those antibodies can cause further problem once the infection is already cured. So that's what we call post-streptococcal disease. This includes the rheumatic fever and also the glomerular nephritis. It's a long word. That's the inflammation in the kidney. Let's talk about each style in each type in detail. The first type is disease attributable to invasion by Aspergillus. It's the beta hemolytic group A Streptococcus. It's on the skin. It's the first disease. This disease type, you don't have to remember these details, but there is one thing you need to know. The puerperal fever. We mentioned this before when we introduced Ignaz Semmelweis, the first doctor who noticed the importance to wash their hands. The doctors to wash their hands. So the puerperal fever is due to streptococcal infection. When the mom gives birth to a baby, and when the bacteria is induced into the uterus, it can cause infection and cause the puerperal fever. And this infection without treatment is sometimes lethal. So long time ago, before the scientific era, without antibiotics, it's very dangerous to have a baby. And a lot of moms, they said that about 1/3 of the moms cannot survive after they give a baby. So mainly due to infection. This is the first type. Second type of disease is due to the local infection by aspergillus and their byproducts, such as the streptococcus sore throat, like this, pharyngitis, so this inflammation in the throat, and also the skin infection, streptococcal pyoderma, like this. It's a local infection of the superficial layer of the skin. Okay. It's a local infection. The third type is due to the production of exotoxins by group A bacteria. Toxic shock syndrome. The exotoxins can induce shock, bacteremia, respiratory failure, and also multi-organ failure. So that's because we are not teaching the clinic courses. We don't want to talk about this in detail. And another disease is called scarlet fever. Scarlet. What's the meaning of scarlet? The red color, right? The special red color. It's this color, okay? This is results from the pathogenic exotoxin produced by the bacteria, the aspergillus. And in the patient, this red color first appear on the body, on the trunk. Then it can spread to the arm and legs quickly with this typical color, the red color. It's not common anymore, especially in recent years, due to the improvement of hygiene condition. But recently, I saw some report online that there are some cases reported in China. Some kids got scarlet fever due to this pathogen infection. And the doctor told me that when he was young, he's about 60 years old. When he was young, he used to treat patients with scarlet fever, but he hadn't seen this for a long time. But in recent years, there are some isolated cases of this disease in China. Okay. The last type of disease is due to the post-infection disease, post-streptococcal disease. So that's not due to the bacterial cell itself. It's that during the streptococcal infection, your immune system produces a large amount of antibodies, okay? And these antibodies can bind to the antigen to form the antigen-antibody complex. And the antigen-antibody complex may precipitate in the tissue, and that can cause inflammation and other problem. The first one we want to introduce is the glow. Anyone read this word? Nephritis, glomerulonephritis. So that's the inflammation in the kidney. How it happens? So that's during the streptococcal infection, your body produces a lot of antibody. This is the structure in the kidney. This is the basement membrane. The green one is the epithelial cell. It's a special epithelial cell in in in, in the kidney. And this red, the yellow one is endothelial cells. Okay, this is a healthy structure. But the antibody, okay, can get into the basement area. It binds to antigen to form the immune complex. What will happen if a lot of antigen-antibody complex forms? It will activate the complement molecule, right? So that it will degrade the tissue here. It may causes damage there. So this is one of the post-infection disease. The second type is rheumatic fever. So that's the most serious problem after infection because it results in damage to the heart muscle and valves. It's a similar way that the antibody can bind to the antigen, activate the complement, and causes damage in the heart muscle and the valve. Okay. Laboratory test. The specimens we can collect the throat swab, the pus, the blood, or the serum. Then we make a smear. You will see the typical structure, the chain structure, gram-positive. To culture the bacteria, we use the blood agar plate, and on the plate you will see the hemolysis. And you can also detect the presence of specific antigen. Okay, the ASO test. Remember ASO, right? Anti-streptolysin O. So this is the antibody. ASO is the antibody. Well, this antibody can recognize streptolysin O, okay? ASO is in the patient's serum. So what you do is that you get the patient's blood, then you get the patient's serum, then you make a serodilution of the patient's serum. Then in each tube, you add in the same amount of streptolysin O, the antigen, and incubate for some time. After that, you add in the red blood cell. The streptolysin O added to the tube will be neutralized by ASO, right? So after neutralization, it's not hemolysis anymore. But further dilution would dilute ASO so that it's not high enough to neutralize the SO you added in. So you will see the hemolysis. And based on the titer, what fold you dilute the patient's serum, you can decide whether it's positive or not. And this is commonly performed to confirm the rheumatic fever. Okay, because rheumatic fever is a serious problem. You can use the ASO test to decide whether your patient is suffering from rheumatic fever. Okay. Okay, to treat this infection, we usually use penicillin G as the first choice. This gram-positive bacteria, penicillin G can kill them. And if the patient is allergic to this antibiotic, you can try other antibiotics such as the macrolides. But we should keep in mind that antibiotics can only kill the bacterial cell themselves, right? If it's the problem due to the post-infection disease, due to the immune production, the antibiotics cannot work anymore. So you cannot use penicillin G to treat rheumatic fever and other post-infection disease. How about we take a break? Okay. Streptococcus pneumonia. So from the name, you know that this is a pathogen which can cause lung infection, right? So that's a major clinical findings. Let me show you the morphology of this bacteria. First, even though this is belong to the genus Streptococcus, but Streptococcus pneumonia usually, okay, here like this, they are diplococci. They don't form long chains, but they form pairs. So it's diplococci in pairs and gram-positive. And you can see the presence of capsule structure surrounding the cells. So they do produce capsules. And the capsules is their important antigen molecules. Let me show you later. And we culture this bacteria on blood agar. But when you culture the bacteria, you want to be careful that this bacteria is able to produce autolysin. Autolysin is the molecule produced by the bacteria itself. It can kill itself. So if you check the colony, you will see that in the middle of the colony, it's depressed, it's lower, while on the rim of the colony, it's elevated. So that in the middle of the colony, that's the older cell, they kill themselves so that the new cells will get more nutrients. So that's due to the autolysis. So it indicates that if we collect specimen for examination, we want to culture the specimen as soon as possible, because after some time, the bacteria will kill themselves and lyse themselves. So you want to keep that in mind. And it's alpha hemolytic. So this is the alpha hemolytic bacteria. Okay, the capsule. We mentioned that the capsule is the important antigenic structure. It contains polysaccharide, and that's based on the polysaccharide types. There are more than 91 different types of polysaccharide. So that's a major antigen. And this polysaccharide antigen can be discovered in the patient's urine or in the CSF fluid. So you can detect the presence of this antigen. And there is a specific test to detect the presence of antigen. It's called the capsule swelling test. Capsule swelling test is you mix the bacteria with the antiserum, which contains the antibody. The antibody can recognize the antigen on the capsule. So the binding of the antibody and antigen is specific, so that after binding, the capsule structure will swell. And you can see the swelling capsule under light microscope. So this is a capsule swelling test. And this test is specific because it relies on antibody antigen interaction, and this interaction is specific. Okay. The pathogenesis, the bacteria can produce this material which contributes to their pathogenesis. First one is the capsule. Keep that in mind. The capsule can prevent or delays phagocytosis. So that's a layer of material covering the cell which can protect them. Phagocytosis. And they produce the pneumolysin. It's a molecule which can destroy red blood cells and also inhibit lymphocyte proliferation and inhibit neutrophil chemotaxis. Do you remember the meaning of chemotaxis? Yeah. So we learned this in immunology that immune cells, such as neutrophil, they can sense the signal so that they will migrate to the site to fight against infection. That's chemotaxis. And the surface protein mediate adhesion, and the bacteria is also able to produce IgA protease, which degrade IgA antibody specifically. The disease caused by Streptococcus pneumonia include first, the most important one, the lung infection causing pneumonia, but it can also cause other problems. Some of them even more severe, such as meningitis. What's the meaning of meningitis? It's a brain infection, right? And also endocarditis. That's inflammation in the heart, and also the septic arthritis in the joint. This is a different type of clinical findings. Just keep these words in mind. We don't want to talk about the detail and also the mechanism. For laboratory test, the specimen we can use sputum, blood, CSF, and keep in mind that all these specimens should be sent to the lab immediately, that they are all producing the autolysis. If it's delayed, you won't be able to find the live bacterial cell. And in the stain smear, you will see the typical gram-positive diplococci. This is a typical morphology. And you can also see many neutrophils in it and many red blood cells. So the bacteria can induce the neutrophil infiltration. And the specific test is the capsule swelling test we explained just now. Culture, we use blood agar, and it's alpha hemolytic. To treat this infection, penicillin G is again the first choice, the first line of antibiotic. And if some strain, some isolate is resistant to penicillin G, you can try other antibiotics such as vancomycin. And it's important to prevent this infection, Streptococcus pneumonia infection. We do have vaccines available. There are two types of vaccines. The first type is the polysaccharide vaccine. So that's the polysaccharide from the capsule. You combine different types, 23 different types of polysaccharide from the capsule and use that as a vaccine. Another type of vaccine is pneumococcal conjugate vaccine. Conjugate means that you conjugate the polysaccharide to a protein carrier. So you link them together. And this conjugated vaccine is more efficient to stimulate immune response. So usually when we give the kids, newborn kids, when they are less than 2 years old, we give them this conjugate vaccine. And for adults, especially the elder people, we can use the polysaccharide vaccine. Did you receive any pneumococcal vaccine when you were little? You don't remember? Do you have any record? Okay. You want to find that? Okay. In Western countries, like in America, the kids would receive this conjugate vaccine. Okay, so it's more effective. While in China, we do have both types. And I would like to recommend the older people to receive this polysaccharide vaccine. You have to pay by yourself, but it's useful to prevent any possible infection. I just received this vaccine a couple months ago. The polysaccharide vaccine. Yeah. Oh no. Each injection will last for 5 years. So you can do that in every 5 years. Okay. So that's the gram-positive cocci. We introduced Staphylococcus and also Streptococcus.